On this week's Arabidopsis Research Roundup, I'm delighted to be joined by Stefan Kepinski from the University of Leeds. And we're going to be talking about a recent open access paper published in Scientific Reports, the title of which is The Developmental and Environmental Regulation of Gravitropic Setpoint Angle in Arabidopsis and Bean. So thanks very much, Stefan, for joining us. And it will be great, I think, if you can give a little description about what gravitropic setpoint angles is. People might not be so familiar. And then how you've worked with that in this current paper.
Yeah, great. Thanks, Graham. Thanks for having me on. So, growth angles are, of course, a tremendously important aspect of the plant's developmental biology because it really determines the plant's ability to be able to capture resources it needs for growth. So, holding leaves out to the sun and spraying out root systems so it can maximise uptake of water and nutrients, etc. And there's a particular class of angle that we're very interested in, and those are, as you pointed out, the gravitropic setpoint angles. And so these are where a branch or any organ is being maintained with respect to gravity rather than to the parent axis or the main stem. So an example is the lateral roots that we're looking at, the lateral shoots, and there's an easy test. If you've got a gravitropic setpoint angle, you just tilt the plant so the branches are moved away from the angle it was at, and if it bends back or affects the tropic response to get back to where it was, then you're dealing with the GSA. Mm-hmm. So they are very interesting because most, as you'll notice from the fact that most of plant life is held out on these branches of some sort. Non-vertical GSAs are very intriguing because when you're looking at a sort of reorientation experiment I described, then those lateral roots and lateral shoots have to be able to bend both with and against the gravity vector. Mm-hmm. So roots bending down and shoots bending up is completely uncontroversial, but to maintain a non-vertical GSA, you have to be able to do exactly those things. You have to be able to bend up and down. So that's why they're intriguing. And in a previous paper, we showed that the maintenance of non-vertical GSAs was dependent on an offset mechanism that counteracts gravity response. And also in that paper, which is sort of the basis of this paper, we showed that Auxin, so if you grow, say, Arabidopsis root system or Arabidopsis plants on Auxin, then their lateral roots can become more vertical. And we also went to look at a range of mutants, and if mutants that either had higher levels of Auxin or greater sensitivity to Auxin, then we found that their root, lateral root and shoot branches were more vertical, and the reverse is true for mutants that are either at lower Auxin levels or lower sensitivity to Auxin. Their branches were more horizontal. And we did a range of other things too to show that all of this was going on, this regulation by Auxin of growth angle was going on in the gravity sensing cells. So this was the starting point for this paper that we're discussing today. We know that Auxin has a central role in regulating growth angle, and so we were interested in whether the different patterns of growth you see when you just look at a root system, the lateral root's born, and then it comes out, it's growing at a quite horizontal angle, and then it becomes increasingly more vertical. So is that pattern of growth associated with differences in Auxin content, for example? And also because growth angles are a very important thing in terms of capturing resources, how do environmental signals impact that? Could we understand whether that was going through the same pathway? So the first part was it's mostly about environmental regulation, but the first thing we did was exactly to try and look at the thing I just mentioned, which is a lateral root emerges at a very horizontal angle and then it becomes more vertical as it grows out. And crucially, going back to the concept of GSA, at any point in that time when it's growing out, you could reorientate it and it would come back to where it was. So it's maintaining a GSA, but those GSA values are different as it grows out. So we asked, knowing that Auxin is making lateral roots more vertical, is it simply the case that there's more Auxin in those older lateral roots? And to do that, we used some Auxin reporters, which are now reasonably well known, but for anybody who doesn't know about them, so these were called domain 2 venous and R2D2. And R2D2 is just a ratiometric version of the original domain 2 venous reporter. And this works as a small motif from an Auxin A protein, which is then fused to the fluorescent protein venous in this case with a nuclear localization signal. And that small section of the Auxin A renders it destabilizable in response to Auxin. So if you've got lower levels of venous signal equals more Auxin, all other things being equal. So when we looked at that, we looked at both actually, domain 2 venous and R2D2. We found that indeed when we looked in those older lateral roots, they seem to contain more Auxin. So these reporters allow you to infer something about the levels of Auxin, as long as the machinery required to do this destabilization is there. So one of the important parts of that machinery in the case of these reporters are the tier 1 family of Auxin co-receptors. So to check that there wasn't some underlying difference between younger lateral roots and older lateral roots, we just made reporters for most of the receptors there, tier 1, FB2 and FB3, certainly the ones that seem to be most important in the root, and just to check there was no difference in sensitivity or capacity to degrade the reporters, and they were the same, so that was good. So there you can see there's quite a neat sort of self-organising thing. The lateral root, when it's quite young, has less Auxin than as it gets older. I'm imagining its capacity to synthesise its own Auxin increases, and it becomes more vertical as a result.
So does the, if I interject there briefly, does the fact it's got more Auxin allow it to bend more? Do you think that's linked in?
No, yes. I mean,
because it's bending more to the vertical as it gets older.
To the vertical, it's not because it's better able to do gravitropism. That's because of the thing that I didn't really talk about at the beginning, the previous paper.
Oh, okay.
So we'd shown that this offset mechanism, so if you think about a primary root just going straight down, that has no offset mechanism, it just goes straight down. But with this offset mechanism, which is what you see in these lateral branches, you can have this non-vertical growth. Now, interestingly, Auxin, as well as doing all the business of driving tropic growth in a Kolodny-Wendt type way, it also has a separate role in decreasing the magnitude of this offset. So, what's happening in this case is we think that the more Auxin being around is meaning that the value of that offset is smaller, and so it becomes more and more like a primary root until, when it's going vertically, it's essentially indistinguishable from a primary root. So that's the idea there. We then moved on to look at environmental signals, and the first one we looked at was the effect of low phosphate. So previously, or about this published while we were doing one of our collaborators, Chris Alverton in Ohio, published a paper saying that on low phosphate they had shown that lateral roots grew more vertically. We'd also seen, and because of all of the work that I've just described, showing that Auxin makes lateral roots grow more vertically, we wanted to ask, was that being mediated through this same family of receptors? So we looked in a tier 1 mutant, and although there's an alteration in the GSA of those mutants, the response to low phosphate, which is becoming more vertical, is still there, and we looked in. So we actually looked at the expression levels of these different receptors, and came to the conclusion that we probably need to be looking at a tier 1 FB3 double mutant. When we looked at that, then we lost this response to low phosphate. So that was suggesting that tier 1 and FB3 are important mediating this response. Looking at low nitrate was the second thing we did, and low nitrate had the opposite effect. It made lateral roots, in Arabidopsis at least, more horizontal. And previous work on looking at the effects of nitrate on root system architecture had implicated this receptor protein AFB3 as being important, and so we looked at AFB3, and that had lost the response as well. So that response at least seems to be going through the AFB3 receptor. Well, at that point, we were slightly puzzled because the phosphate result was not quite what we were expecting, because Jonathan Lynch's group in the States, produced lots of work on the effects of low phosphate on the adventitious roots or basal roots in bean, that suggested that at low phosphate they became more horizontal, which makes a lot of sense because in low phosphate soil, phosphate's not very mobile, so most of the phosphate is in those upper layers of the soil, so that makes a lot of sense. So we then, the reason for this paper being titled In Arabidopsis and Bean was because we then looked to look at bean to see what was going on.
Yeah.
And sure enough, when we looked at the response of these bean adventitious roots, these basal roots to low phosphate, they were going up a bit. And interestingly, when we low levels of Auxin treatment, they also went up. So this is the opposite of what happens in Arabidopsis lateral roots.
Oh, okay.
And when we increased the amount of Auxin that we were growing these beans on, then eventually their roots became more vertical. So it's an interesting difference between the response of these basal adventitious roots to Auxin and lateral roots. Because when we looked at the lateral roots in bean, they were just like the Arabidopsis ones.
Okay.
They just became more vertical. So that was interesting because it suggests there's some sort of difference in response in those different classes of roots.
Mm-hmm. Okay.
And then the final experiments we did were in the shoot, where we looked at whether shading, reduction in light quantity, and changes in red far red ratio and high temperature could affect branch angle. Mm-hmm. And we found they did. So under shaded conditions, we got branches, so these are the cauline branches in Arabidopsis, became more vertical, which makes sense, I guess, because of part of the the idea of shade avoidance is to escape the shade of your neighbours, and so reducing the amount of lateral spread of your growth is a good idea, I guess. We tested a mutant called PIF4 because PIF4 has been shown to be important in mediating shade avoidance responses, and slightly interestingly, it didn't seem to be doing anything in the context of this response. So we still got the change in angle, the shift to a more vertical growth angle in the PIF4 mutant. Okay. But I was probably saying now there are lots of other PIFs to be looking at. So I think that's more likely a redundancy question thing than anything else. And then finally we looked at high temperature. So high temperature induced more vertical growth. And one of the reasons for looking at high temperature is that a long time ago in Mark Stell's group, they'd shown that at high temperature Auxin levels were increased. And so it was another way of testing this idea. And again, we looked at high temperature at 28 degrees, we find out those root branches those shoot branches are growing at a more vertical angle. And again, this is something that's independent of PIF4, and PIF4 had also been implicated in high temperature responses. So that is, and that was about the side of it.
Yeah, okay. So you've shown previously, I think, is that right, that the Auxin is involved in the GSA and the shoot as well, or is that something? Yeah, okay. So the mechanism, the fundamental mechanism, seems to be the same in the root and the shoot. Okay. So this is really just moving on that and confirming that these are the sorts of things you would have expected to be happening, knowing that Auxin is doing what it's doing. But yeah. Okay. So just a final question. You kind of touched on this, but thinking transitioning this work into the real world, let's say, and the difference between phosphate and nitrate. So plants or the roots will grow out when there's less nitrate, but they grow down when there's less phosphate. So what's the real reason for that? Do the plants know that the nitrate will sink more will stay in the upper area, and the phosphate will sink?
So this is the paradox, if you like, if you want to call it that.
Mm-hmm.
They're doing entirely the wrong thing.
Right. Okay.
In this case. Right. So a good idea. Again, this is based on the work of Jonathan Lynch, who are big fans of his work. Mm-hmm. He has this concept of stick, cheap and deep as an idea type for a root system architecture to deal with low nitrate, and in fact drought in dry soil conditions. Mm-hmm. So what Arabidopsis is doing is the opposite of what is a sensible thing to do in that case. Now, that's, you can argue about whether it would really apply to this small weed that is good at growing, short life cycle, good at growing as weeds do. But it's something to bear in mind. One thing we need to do, and we're doing, is looking in other species. The other thing so the nitrate response is not what you should be doing, and the phosphate response is the opposite of what you want to be doing as well for lateral roots. But it's probably the case that in the context of a larger plant that has more adventitious roots and relies more on the adventitious roots, then the responses that we've seen and that the Lynch group have reported make a lot of sense. But it's something to be borne in mind when thinking about manipulating architecture to improve crop performance. That's the thing. So this gives some insight into the mechanisms that are going on there. And if you want to change, if you do want to try and shift root growth to more vertical, deeper root systems, then these are some of the considerations that can be borne in mind.
Yeah, exactly. So whereas the Arabidopsis example might be slightly different from what you might expect in other plants, the fundamental mechanism that controls how the root grows is obviously present there in the Arabidopsis.
That's what we think, and that's what we're doing. We're working in wheat, and we're actually working in a range of species now to examine this more closely.
Excellent. All right. Well, thank you very much, Stefan, for explaining this paper in some good detail there. And as I always say, we look forward to some extra work in the future. Certainly.
Great. Thanks very much.
Thanks.